to understand this response, it helps to know something about leak gas exchange. I may take a tangent, talk a little bit about measuring stomata conductance and photosynthetic with gas exchange systems. But the general response of stomata to CO2 is that as the CO2 concentration rises, the stomata tend to close. And as the concentration decreases, they tend to open. This is a gross generalization because some plants have stomata that don't respond to CO2. And in some species of plants, there's not a CO2 response unless the plants aren't water stressed. So it's not as ubiquitous as the stomatal response to light, which all stomata respond to light of one form or another, but not all stomata respond to CO2. Most stomata respond to CO2, but not all. Interesting thing about the CO2 response is even if you have a plant in the dark with closed stomata, if you decrease the CO2 concentration enough, the stomata will open in the dark. So what does that imply for where the energy comes from? To open the stomata, right? Remember what happens. First thing that happens is protons get pumped out of the guard cell into the apoplast. And that takes energy. Where does that energy come from? In CO2 response. Not from photosynthesis, not from photophosphorylation. Can't come from that, so it must come from something else, like
Sucrose,
respiration. Yeah, sucrose and breakdown of sugars in respiration, glycolysis, tricarboxylic acid cycle, and oxidative phosphorylation in the mitochondria. So guard cells are loaded with mitochondria as well as chloroplasts, usually. I mean, they're full of mitochondria. Usually they have lots of chloroplasts too, but they do have mitochondria, so presumably energy can come from that. And there's not a lot that's known about the CO2 response of stomata. In particular, the mechanisms of that response aren't known. How the guard cell senses the CO2 and what biochemical steps precede the extrusion of the proton across the plasma membrane. That kind of stuff isn't very well known. But a little bit is known, and one thing that's known is the CO2 concentration to which guard cells respond is not the CO2 in the air, it's the CO2 inside the leaf. It's the CO2 concentration in the air spaces inside the leaf, otherwise called the intercellular CO2 concentration, usually symbolized by a C with a subscript I, CI. So as I said, the mechanism isn't well known, but some interesting things, like if you remember from this slide from last time, CO2 sensing by Rubisco is written on there, and it doesn't say much about the mechanism, but this statement is found in Taez's book, this one. It is likely that rates of CO2 fixation by guard cell chloroplasts can regulate zeaxanthin concentrations and integrate light and CO2 sensing in guard cells. So it would be interesting to me to take those orchids, Paphiopedilla, and see if they respond to CO2. They don't have chloroplasts, remember, they don't have chlorophyll. They respond to blue light, but not red light. Do they respond to CO2? I don't know. Be an easy experiment to do. Maybe we should get some orchids and try it. I've got a gas exchange system. You want to take me up on it? See me after class. Okay. Anyway, so there's not a lot known about CO2, but some things are known. So here's a general response. This is from Wayne Polly and some of his colleagues in Texas. they had a series of tunnels to look at effects of elevated CO2 on ecology of prairie species in Texas. And what they would do is have a long tunnel, and they'd add CO2 into one end, and had an airstream that blew through the tunnel and came out the other end. And they could measure rates of photosynthesis by looking at the drop of CO2 along the length of this tunnel, and did things like that. They also did leaf level measurements and things. But anyway, here's this general response of stomatal conductance to CO2 concentration. And you can see C3 forb, this thing, C3 grass, C4 grass. In all these cases, as the CO2 concentration increases, the stomatal conductance decreases. All right, so the internal CO2 concentration is an important parameter, right? So here's this simple equation for net CO2 assimilation rate, this Ohm's law analogy, right, of the flux of CO2 into the leaf is some constant of proportionality times the driving force. And in this case, the driving force for CO2 moving into the leaf is the difference in concentration between the ambient air CO2 concentration and the concentration inside the leaf. That difference in concentrations is what's making the CO2 move into the leaf. And the factor that relates those is the total leaf conductance for CO2. Remember, that's the combination, parallel combination of the two surfaces of the leaf. And each surface is the series combination of the boundary layer conductance and the stomatal conductance. So this is, I should write this with, like I do here, with the approximately equal sign. Because if you think a little bit more about what's happening, not only, when the stomata are open, not only is CO2 diffusing into the leaf, but water vapor is coming out. And so the diffusion of CO2 in depends somewhat on how much water vapor is diffusing out at the same time. Because those CO2 molecules have to negotiate all the stuff that's going on there, right? And so a more appropriate equation, I mean, we can rearrange this equation to solve for CI, right? CI equals A divided by the total conductance. Minus sign, right? Something like this. Right, we can solve that for CI. But that's only an approximation, and a better equation accounts for the transpiration rate of the leaf, of how much water, you know, what's this influence of this stream of water molecules moving out of the leaf as the CO2 is trying to move in. And so this equation is more appropriate. And if you make a measurement with, for example, a Lycor 6800, now it's the latest model, this is the way that the internal CO2 concentration is calculated inside a machine like that. Because, you know, I mean, how are you going to measure that concentration inside the leaf? In the Lycor system, they use an infrared gas analyzer to measure CO2 concentrations. It's a big thing. Well, now maybe it's like this. You can't stick that inside the leaf and measure a concentration. It's too small. How do you measure that concentration? Turns out you don't measure it, you estimate it based on the total leaf conductance for CO2, the transpiration rate, the ambient CO2 concentration, and the net photosynthetic rate. How do we know this equation is appropriate? That was my question, right? Okay, I just told you, yeah, that's the equation to use. How do you know that's right? How do you know it's right if you can't measure that CO2 concentration inside the leaf?
Imagine how much decrease in the air.
maybe, I don't know, maybe you could do it that way. You could, like an elaborate sort of fitting exercise and figure out, okay, if you can measure this, and this accurately, then maybe, you solve it by, yeah, maybe, I don't know. There's another way that I've seen how people do it. So for this, we have to talk a little bit about gas exchange, right? So one way to measure photosynthetic rates or stomatal conductances is to put a leaf, actually put a part of a leaf inside a small chamber and measure the CO2 concentration going into this chamber and the CO2 concentration coming out of this chamber. The difference between these two, okay, so if the stomata are open and the leaf is photosynthesizing, if we put in like 400 ppm CO2, air with 400 ppm CO2, why 400?
Standard.
Standard. What's the CO2 concentration outside on a day like today? It's about 400. All right. What's the CO2 concentration inside this room? It's way higher than 400. And the longer we stay in here, especially with the door shut, the higher it's going to get as the sleep even. It's true. It's true. So I don't know what the CO2 concentration in here is. Maybe 600-700 parts per million. Maybe higher. Maybe it's 1,200. So, anyways, if we have an airstream going in here with 400 ppm CO2, if the stomata are open and the plant's photosynthesizing, some of that CO2 goes into the leaf, and so the CO2 concentration coming out may be 380 parts per million. This difference in the CO2 concentration is related to the net CO2 assimilation rate. And if you know something about the total leaf conductance for CO2 and some of these other things, we can calculate a photosynthetic rate that's appropriate for this difference in CO2 concentration and get a handle on this term, A. Similarly, we can measure the water vapor concentration going in and the water vapor concentration coming out. And if the stomata are open, they're going to add water to that stream of air. Water's coming from inside the leaf and diffusing out, the transpiration. And so,
wouldn't transpiration be species dependent though?
yes, it's photosynthesis, and they're both depending on all the environmental conditions that you put on that chamber. Yeah. Depends on species, depends on their environmental history, depends on how much nitrogen's available to the plant, it depends on the light intensity, depends on the VPD, it depends on all of these things. Yeah, it does. That's why people like to use this type of gas exchange system, which is called an open system, versus a closed system where you put a leaf in the chamber, or a transient system, put a leaf in the chamber and watch the CO2 concentration in the chamber decrease and the water vapor concentration in the chamber increase at the same time. And you can estimate photosynthesis and stomatal conductance based on a closed system too, a transient system. That's how the Glycor 6000 and the Glycor 6200 systems work. But with the 6400, they went to a closed system, or an open system. Because in an open system like this, you can keep those environmental conditions constant in the chamber for, theoretically as long as you want, and make your measurements. Because you're right, the rate of photosynthesis, the transpiration rate, all that stuff depends on the conditions. And in a transient system with the thing closed, those conditions are changing all the time. So, for that and other reasons, the gas exchange systems that you can get these days are typically full. Okay. All right, but from this difference in CO2 concentration and water vapor concentration, we can determine the transpiration rate and the net CO2 assimilation rate for the leaf inside our chamber. From those, okay, so from the transpiration rate, then, if we use an equation like, or similar to this, the total leaf conductance for water vapor times the saturation vapor pressure at the leaf temperature minus the vapor pressure of the air divided by the total pressure from measuring the transpiration rate. We need to know the leaf temperature and some other things. We can calculate a total leaf conductance for water vapor. From this total leaf conductance, if we know the boundary layer conductance, we can calculate this the model conductance for the leaf. So, by making gas exchange measurements, we can quantify all of these factors and use those estimates to get a value for CI that's appropriate. How do we know it's appropriate? This is my long-winded way of introduction. How do we know it's appropriate? Well, there have been some clever experiments done. All right? Okay, so let's say we have a C3 leaf in here. A leaf from a C3 plant. What's an appropriate internal CO2 concentration for C3? Do you know? C versus C4. Which one's higher? Which one's lower? Okay, I'll give you a hint. If the ambient is 400, they're both going to be less than 400. Which one's more or less? Which one is lower? CI for C3 or CI for C4? And the next question is why? Okay, well, C4 is lower than C3. The ambient on a C3 on 400 ppm, or the CI on a 400 ppm ambient for C3 may be 280 parts per million. 280 ppm. And for a C4, maybe 100, let's say 110, close to 100. Yeah.
Did you say that was ambient or internal?
This is internal. On a 400 ppm ambient, let's bump it up a little, because this will be consistent with my numbers later. 160. Why the difference? Do you know?
Because of the way C3 plants fix carbon, C4 plants are more efficient internally, but they also go through more steps to fix that carbon.
And more efficient. Okay. How or why are they more efficient?
Because they're
It's like a comprehensive exam, right?
no, it is, and I have a question from that that regards this same thing.
Uh-huh.
Because in C4, the PET C or the C4 equivalent of the Visco doesn't get bogged down by added carbon dioxide or added oxygen.
Right.
So
Right.
if oxygen concentrations were higher, then C3 would be at a disadvantage, where if carbon dioxide concentrations are higher, then C3 would be at an advantage.
Yeah, yeah.
So
Pretty much.
suggesting with
Pretty much.
this
Um,
global climate change, that C3 would be, that C3 would, be at a greater advantage than C4. We think that C4 are more water efficient, but if global climate change is due to CO2 or heavy carbon containing molecules, that would give C3 better advantage.
yeah.
Is that flawed?
No, I mean, I would have said things a little bit differently, but in general, yeah, what you said is right.
So long as they aren't starving for water.
Or nitrogen.
Right.
Yeah. Okay, but the one thing that I would, that caught my ear was C3s and C4s both have Rubisco, and they both have Calvin cycle, they both fix carbon exactly the same way. But the C4s have a front end on things.
Yeah.
Before you get to that Rubisco step. And what that does is it concentrates the CO2 around the sites of where Rubisco is. So that oxygen doesn't have much of a chance of competing at the active site of Rubisco. The way they do it is, like you say, the initial CO2 fixation in the C4 is catalyzed by PEP carboxylase.
Yeah.
And that enzyme only gloms onto CO2. Oxygen can't do anything to it.
Yeah.
So it's insensitive to oxygen concentration. So in the C4, PEP carboxylase gets that CO2, fixes it with PEP, with phosphoenol pyruvate, a 3-carbon thing, to make a 4-carbon molecule. So hence the name C4 is the initial product of that carboxylation. That C4 compound, malate or something, moves from the mesophyll cells to the bundle sheath cells in the C4. Remember, C4 anatomy around the vascular bundle has a group of cells called bundle sheath cells, and they're very important in photosynthesis because what happens is that malate goes to those bundle sheath cells, gets decarboxylated, right? So the CO2 comes back off of it, and then the 3-carbon thing left moves back out to the mesophyll to cycle around again. And by doing that, the CO2 concentration within those cells is really increased a lot over what's happening in the mesophyll cells. And that essentially overcomes the competitive inhibition by oxygen at the active site on the Rubisco molecule. And in the bundle sheath cell, then Rubisco fixes CO2 in a Calvin cycle, blah, 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 just like the rest of it. So by doing that, since in the C3 plant, though, Rubisco is the initial carbon fixation step. And oxygen competes with Rubisco. What's more, that oxygen, for every 2 oxygen molecules that are fixed by Rubisco, 1 CO2 molecule comes back off. And that's called photorespiration. It's a complicated series of steps that takes place between a few different organelles. But anyway, because that oxygen fixation by Rubisco ultimately releases some CO2, then those kinds of plants, C3 plants, can't lower the CO2 concentration very low in their intercellular air spaces. Because the lower they get, the more oxygen can go on there, and then it makes more CO2, right? And so that photorespiration is negligible in C4 plants, but it's appreciable in C3 plants. So that's why these difference in internal CO2 concentration. Sorry, long-winded way of taking a break. How do we know this equation is right for CI? Okay, so you clamp on the leaf, you measure something, and the machine tells you, oh, my CO2 concentration inside the leaf is 280 parts per million. How do you know that's right? How do you know to believe it or not? Well, there's been some clever experiments. Okay, so let's say we have a C3 leaf in here, getting back to this. Let's say we have C3 leaf in here, and we've got 400 parts per million coming in and whatever coming out. You know, the concentration coming out is going to depend on the flow rate, it's going to depend on the photostatic rate, it's going to depend on a lot of things. But anyway, let's say that we have it. We can calculate a rate of photosynthesis, the rate of net CO2 assimilation. And this rate is greater than 0 for a concentration of 400. But what if we put 300 in? And we get 285 out. Well, we still have a positive photosynthetic rate, but it's smaller than it was before. Let's say that the internal CO2 concentration in this leaf is what I say here, 280 parts per million. What if we put in 280 here? What are we going to get out? We still might get 285 out. Or maybe we get 280 out, right? If we put in 280, we're going to get a photosynthetic rate close to 0, right? Because we're at the CO2 compensation point. In other words, if we make a graph, we can make a graph like this. I use the black marker. We can make a graph of CO2 assimilation rate and CO2 concentration here. And let's call this CI. We get a graph something like this. Actually, what it'll look like is we'll get a small response here. Something like that. And this value right here is the CO2 compensation point, right? The rate at which the net CO2 assimilation rate is 0. Whatever that CO2 concentration is, that's what CI. All right, so here's what you do. A long setup, right, for a short payoff. People have made these gas exchange systems in such a way that let's have two systems, one on each surface of the leaf. All right, so this is my leaf in here. Right, the leaf is sideways. We have one chamber on the top of the leaf, one on the bottom of the leaf. And you measure the CO2 going in, CO2 concentration going in, and the vapor pressure going in and coming out on both surfaces. So you can calculate separate rates of photosynthesis and transpiration on the top surface or the bottom surface. Now, to do this experiment, you're going to have a leaf that's hypostomonic. I mean, that's anti-stomonous, right? Stomonic on both surfaces. And it would be good to have about equal numbers on both surfaces. Okay, so here's what people have done. Set this up. Let's say we have 400 parts per million coming in, 380 coming out. We calculate a photosynthetic rate on the top of the leaf, whatever it is. And from these data, we can calculate a CI from the top of, let's say, 180 parts per million. Okay. And using this equation and the stuff that we measure, we get a CI of 280 inside this leaf based on gas exchange from the top. Then on the bottom, what we can do is vary the CO2 concentration until we get 280 going in. Well, here we go. Here's how you do it. You measure this, same conditions on the bottom. Start at 400, whatever it is. We calculate a photosynthetic rate for the bottom. Now, start decreasing the CO2 concentration here, like this. Decreasing that CO2 concentration until you get to a photosynthetic rate of 0 on the bottom. Which means that the ambient, the air CO2 concentration is the same as the CO2 concentration inside the leaf, because there's no diffusion, there's nothing, nothing to move the molecules in there. What is that CO2 concentration? Turns out, 280. It's the same thing that you calculated from the top conditions using this equation. That gives good confidence that that equation is correct. So if you're interested, guy in Wisconsin, Tom Sharkey, he's done a lot of photosynthetic work. He's got a paper talking about this. Anyway, you don't just have to take my word for it. You can do an experiment and prove it to yourself. And this is how you do it. We'll come back to some of this gas exchange stuff. That's what I take time to do it now. Okay? You got an extra $35,000 sitting around, you can buy a gas exchange system yourself. Play around at your leisure. Okay. That's about what they cost. Okay. Now, here we go. So, under favorable conditions, as we talked about a minute ago, the internal CO2, plants regulate their small conductance so as to keep the internal CO2 concentration about constant, believe it or not. And favorable conditions highlight no water stress, moderate vapor pressure deficits. Plants tend to maintain a constant relationship between the ambient CO2 concentration and the internal CO2 concentration. So that ratio of CI divided by CA. For C4 plants, CI is about 0.4 of CA. For C3 is about 0.7 of CA. And the difference is because of, as you explained with the fixation